Assalamu alaikum. Welcome everyone for today's session. So today we have with us Dr. Mahfuzur Rahman. He graduated from Bangladesh Agriculture University in veterinary science. Then he did his Ph.D. in Kagoshima University, Japan. After that, now he is working in the University of Wisconsin Madison as a postdoctoral research fellow. So today, we'd like to invite Mahfuzur Rahman to start his presentation. Mahfuz, you can please share your screen and start your presentation. Thank you.
Thank you, Md, for your introduction. Yeah, I'm trying to.
Can you also your video?
Yeah, sure, I can. Just a minute. I hope everybody can see my presentation and also my face.
Yes, thank you.
Okay, so hello everybody. As Moinbhai said already, my name is Mahfuz. So this study is one of the part of my doctoral study, and I'm going to introduce this study to all of you. So I may have some small background in the beginning. So this is the research we did I did in Japan, and there is a couple of objectives for this research. First of all, the one objective is to study the dog cancer research, to improve the dog cancer research, and the other one is to improve the human medicine, to contribute to the human medicine. And so from the beginning of the medicine history, we know that several animal models were used to develop the different vaccines or drug. Like this picture shows that like a smallpox was discovered in 1902 from a cow. And from that time, right now, several animal models like mice, primates, dogs were used as a animal model to develop the drugs or to enhance the clinical trials in human medicine. However, very recent study shows what we did in our weight bench or preclinical study, only 8% can translate to the human study. So there's a big lackings in our research or in our animal model. So that's one of the reasons scientists try to focus on different cancer model. They search on the most specific or most comparative or most relative cancer model. That's one of the objectives of our study. So dog was used as a cancer model very early age of the research, and because they have a very reflective life cycle, and also they have the anatomical similarities as shown in here. And also the recent high throughput sequencing from the dog genome, it shows that like 92% of the DNA are common with human, and also they have the respective cancer like human. They have the brain cancer, melanoma, leukemia, and so on, as the human also have. And also they share the common environment, common food nature. So they are maybe the most relative comparative model of human. So I studied the several cancers, but I show here in the canine melanoma because this is the most common canine oral malignancy, and it's consists of like 7% malignant tumors in dog, and also they have the more anatomically human also have the oral melanoma and several other melanoma as like the dog has. So, and also if we want to find the similarities, and if we want to increase our treatment regimen, so we need to, first of all, we need to find the targets, and that's why one of the strategy is to study the central dogma. In our study, we study the transcriptome, that is the middle part of the central dogma, and there's a couple of reasons we compare the human and dog transcriptome, because in our two of our previous studies showed that like we did the small RNA sequencing from dog melanoma, and we found that like half of the differentially expressed microRNA are similar between dog and human, and the microRNA, those are not similar, is actually the sequence are not similar between dog and human. That's the one reason. But like as you see, like 50% small RNA or microRNA are similar between dog and human melanoma. And also one of our previous studies showed that some small RNAs or long non-coding RNA derived small RNAs have also the similar expression between dog and human. So after this, we are thinking that we maybe dog and human transcriptome may have more similarities, like they may have more gene expression or transcriptomic or signaling pathways they share in the same way to develop the melanoma. So one of our hypotheses is the transcriptome analysis of the canine oral melanoma may reveal the global gene expression similarities between dog and human. And so during this process, we need to find out the to identify the gene expression aberration in the canine oral melanoma. And also we need to find out the functional similarities of gene regulation between the two species. So so to achieve our goal, we did the next generation sequencing of the melanoma tissue samples from dog. What we did, we collect the controlled oral tissue from the control dogs and also the melanoma tissue from the clinics. We put it on the RNA later, keep we isolate the total RNA, and we did the next generation sequencing. And after getting back the sequencing reads from the sequencing facilities, we analyze our data ourselves and by the CLC genomic workbench to define the differential expressed genes in canine oral melanoma. And next part, we also did some in silico analysis. In that part, so this is our data, and we draw several data set from the SAI database that are human data. And we compare this canine oral melanoma data with the human melanoma data and also the prostate cancer data. We use the prostate cancer data for our negative control. So during this analysis, we use CLC genomic workbench, Biomart, and several couple of bioinformatic tools. And also we confirm our result finally with the qPCR. So the initial analysis of the sequencing rate shows several upregulated and downregulated genes. The red dot, so this is the Walken plot, and the red one dot means one gene, and the red are the significantly differentially expressed genes. So now, as you can see, there are like more than 1,000 differentially expressed genes in upregulated and downregulated group. So there is a huge number of genes. So we need to sort out those huge number of genes to find out the key things. So from these differentially expressed genes, we first identify the on genes. So there is 80 on genes. That on genes means in the control group, there is no expression, and in the melanoma group, there is high expression. So what is the high expression is like more than 5-fold. And we also set a statistical threshold, like FDL should be 0, and the RPKM or RPKM or FPKM. So RPKM is that like they have some statistical issues that define the count of each gene. So we define that so the on genes should be more than or less than 1 RPKM. That means they should have highly expressed in the respective groups. That means the on genes should be highly expressed in the melanoma, and off genes should be highly expressed in the control group. So by this way, we identify 80 on genes and 241 off genes. And so after identify the on and off genes, we are thinking that there may be some correlation in the chromosomal location, because there are some similar gene domain or protein domain exist in our previous analysis. Oh, I didn't show him here. So when we merge all of these differentially expressed genes in the chromosome, we found that in chromosome 9, so this is the peaks. This peaks means expressed genes. For example, like in NGFR, these genes, if expressed, they have high peak. If don't express, they don't have peak. So we found that the chromosome 9 have consistently increased some on genes. Like you can see here, here, and here. These are the melanoma samples, and you can see these peaks are always present in the melanoma samples, whereas they are absent in the control group. That means that maybe chromosome 9 is very important for the melanoma development or melanoma progression. And we also similarly investigate the downregulated genes. And you can see that similarly, the downregulated genes are highly expressed in the chromosome 1 in the control group in different location, and also they are absent in the melanoma samples. It indicates that there is some hotspot in the chromosome for the melanoma progression, and maybe the chromosome 9 and chromosome 1 can be a good target to investigate. So next we analyze the predisposing factor. We know the transcription factor. I think the transcription factor are the factors that are influence the gene to transcribe. So investigate the upregulated. So we investigate the transcription factor for the upregulated genes, and we found that NF-kappa B and ATF1 are the significantly enriched transcription factor in our upregulated genes. Also, we also find 4 unknown motif in the. They have the same unknown motif in the most of the upregulated genes. We didn't investigate, but it indicates that there is some specific way or specific signaling pathways that trigger the upregulated genes when the melanoma progress. After that, we investigate the oncogenic similarities between dog and human. what you did, we first of all, we compared the differentially expressed genes between our set and the melanoma tissue data set that were drawn from the SI database. We compared the upregulated and downregulated both groups. Interestingly, you can see in the upregulated group, so there is 88% genes are commonly upregulated. So this is the fold change. You mean the genes that are twofold upregulated between dog and human, they have the similar upregulation, and each line indicates one gene. So from this graph, you can clearly see that like 88% genes, like most of the genes, have the similarly expressed between the groups. So when we compare the genes between these two species, we need to I need to mention here because to compare the genes between the two groups, we need to take account the ortholog sequence, so that is one of the reasons the number of genes is low. Like we found like 500, around 500 genes to compare between the species, even though we have more than 1,000 upregulated genes. And then we compared the cell line and our negative control, and we did a cluster analysis. So when we did the cluster analysis, you can see this is the prostate cancer cell data, and that they are clustered separately. And these are the human melanoma cell line. So, and HM tissue means they are the human tissue melanoma, and DM means dog melanoma, and HM the human melanoma. So from this cluster analysis, it's clearly shown that some of the dog melanoma tissue have similar gene expression with the human melanoma because they are in the same cluster. And even though they are more similar than the human melanoma cell line, so it indicates that it indicates that the melanoma has some specific genes in the both species, which triggered the melanoma progression. So from our analysis, we define, we propose 429 melanoma-associated gene signature, in which 284 genes were common between the human and dog, and some other genes are common between the cell line, human cell line, and dog tissue samples. So after finding these common genes between the two species, we are interested to know that which signaling pathways are important to upregulated or to enhance the expression of these genes. So we analyzed the gene signature pathway, GSEF, so gene set enrichment analysis. And we found that the cytokine receptor, node-like, and JAK-STAT are the most enriched signaling pathways among the common genes. So the JAK-STAT is very known pathways in the melanoma in human, and also we find that we know that the melanoma in human are very immunogenic. That means they have more cytokine. So it shows that they have the cytokine. That's the reason they increase the immunogenicity. and they also the JAK-STAT. So we analyzed the fold change of these leading edge genes. The leading edge genes means the genes that that contribute the score of to find out these pathways. So we analyzed the leading edge genes fold change between the two species, and we found that the leading edge genes also have the similar type of expression in between the two species. So it indicates that these three pathways are most important in both species to develop the melanoma, and also they may be the target to make some therapeutic regimen. So from our study, we first report the comprehensive RNA-seq in canadenal melanoma, and we also showed that dog is a good model for human melanoma study and also for the clinical trial. And from our melanoma signature genes, we found that the almost 10% melanoma signature genes are already reported in human melanoma database. And also we showed that the leading edge genes in the common signaling pathways are conserved between the two species. And in conclusion, we portrayed the first comprehensive analysis of dog transcriptome by NGS, and we found that the similar transcriptomic expression were found between the species. And among the common transcriptomic similarities, about 88% upregulated genes, ortholog upregulated genes are common between the human and dog. That's all of this my presentation. And if you have some questions, feel free to ask. I'm very happy to discuss.
Thank you very much, Mahfuz. It's an outstanding research work. Before asking the open question, I'd like to ask you one question.
Sure.
So you checked the dog melanoma. So
right.
How you diagnose this dog melanoma? Is it means you induce the melanoma or it's a wild type?
So these are the spontaneous. So the dog has also developed the melanoma as like human developed. So this dog came to the clinics for the treatment.
Okay.
And they did the surgery, and we collect the tissue.
Yeah. So in our human research, so when we use as a model for any rat model or any dog model or any model, so we need to induce the tumor or something.
Yes. Yes.
So what will be that? It means you check the RNA-seq, everything is in wild type or spontaneous tumor.
Right. So that's why I'm discussing the spontaneous model in the beginning, because in the induction model, we know this is melanoma, and we induce, right? We don't take account the influence of the environmental factor and also the other factors. But in the spontaneous model, we can this spontaneous model have also the influence of the environmental factors. So that's one of our conclusion, that the spontaneous model may adopt more suitably than the induced model.
Yeah, I understand, but how available this? Because means if you want to do any research on melanoma, Means how can you?
Yeah, so
Means,
that's the shortcomings of this or this type of study. So we collect these melanoma tissue samples about for 3 years, 2.5
yeah,
years, I think.
I understand.
Yeah. So, and the sample size is not too big. Like we study the, you can see in here, we study the 8 melanoma samples in our NGS and 3
Yeah.
controls. We can increase the controls, but that's fine. But we only did the NGS for 8 melanoma samples. And overall, we have 17 melanoma samples. So is that this is the most shortcomings.
Okay, so now I would like to everyone. So if anyone have any question, please unmute himself or herself and ask question. We have also in the some question in chat box, but we would like to read later. But please ask any question. Thank you.
Hello, can you hear me?
Yes, I can.
Nice presentation, Dr. Mahfuz,
Thank you.
and well done. So, first of all, we know the melanoma, human melanoma is a pigmentation.
Yeah.
Okay, so it's a black pigmentation. So
Yeah.
there are some kind of lesion which can be bigger, which can be smaller, which
Right.
can be malignant, which can be not. But the malignancy rate is so much less. My question is, uh, if the gene expresses higher or if the gene is upregulated in 9 dog, so in the malignancy time it also expresses higher, or it's the burning question for the next research.
Okay. Actually, we try to find out, like some of the dogs in our study group cohort, some of the dogs have metastasis in the lung, but we don't find any specific genes for the metastasis that are related to metastasis. And I think the size of the cohort is not good enough to compare between the metastasis and non-metastasis.
Okay, thank you so much. And
But
I have
of course,
another
there may
one.
be something.
I don't know if it's the right time to ask it or not. So if someone wants to do that kind of research, so what do you suggest for the next person? Like for the gene expression, they should use the mm, dog, or it's better to use the mice.
Okay. So first of all, we can use the mice. But if we use the mice, we will lose the environmental factors and the other factors that have influence for in the spontaneous model. So if somebody want to study in the metastasis and non-metastasis related genes or factors, they definitely need a large cohort. And for that reason, I think a national project should require.
Okay,
Otherwise,
I have another issue about that.
yes,
Do you check any kind of transcriptor factor for these upregulated gene?
transcription factors.
Yeah.
Yeah, I did the check the transcription factors in the upregulated genes, and as I showed, uh, maybe here, we found some interesting unknown transcription factors. You can see. Uh, so these are the so NF-κB and ATF1 are the most uh,
Mhm.
common transcription factor in the upregulated genes, and there are other transcription factors motifs that are present in the upregulated genes, but we didn't investigate further.
Okay. Okay, thank you so much. Thank you.
Does anyone have any question? I think Mahi, you have some question in chat box.
Oh.
Would you like to ask yourself, or I will ask? Okay, the question is, was it single or pair in data?
Paired in data.
So also genes are significant under if FDR equals 0.
Yeah. Actually the FDR 0 means there are it's actually not 0, but it's like 0.1000 like there is something 1 in the last, should be. So it's 0 means not 0. So and there is, of course, there is some differential express genes.
So in that case, my suggestion, if you put in like that, mathematically it's very wrong. So I think you can write that FDR less than 1 or less than 0.0000 something. So the equal 0 is confusing.
Is it equal? Okay. Yeah. Oh, okay. Yeah.
And the last one question is, I wonder if the study is well powered given you have only 3 control sample.
Yeah, this is one of the question asked by the reviewer, and that's the one shortcomings. We submit this paper in the scientific report, and they came up with these issues, like we have only 3 samples, and we said we analyzed this data with the different statistics, but they are not satisfied. But truly, I think they are well powered, because now there is some statistical model that can also compare the one sample in the RNA-seq.
Okay, that I don't know how you can measure the one sample RNA. Hopefully, some expert can discuss with the more.
And also we confirmed this, like the differentially expressed genes in the qPCR sample, and by qPCR, and there is more samples. Like I said, there is I think 10 controls and 17 melanoma by qPCR. So I think it's fine. 10 is fine, more like enough to have the
It's okay. So yeah. So and the next comment we get from Sanal Kobi is, great outcome, brother. Thanks for the nice presentation. Yeah, that's all you have. So thank you, Mahfuzur Rahman.
Yeah.
And if no one have any question, then we'd like to end our session today. So thank you everyone for joining this presentation. We hope we will have the next session in the next week. I will invite everyone. And now, thank you for today. Bye.